So we are going to talk about the electron transport chain now. So we've just talked about aerobic and anaerobic respiration. And now we're going to bring it into the last leg. And, who cares, right? It's just the very end of it. It's the point where we make like all the ATP. No big deal. All right. So this is pretty much going to be all about the mitochondria here. And so the first thing is you're going to get a very simplified representation of the mitochondria. So it has two membranes. So it's a very interesting organelle. So it has an outer membrane, and you'll never guess what we call this inner membrane. Oh, shoot, I gave away the name already. And so inside of the inner membrane is where, so Krebs cycle means citric acid cycle. They're just two names for the same thing. That's where all of the previous stuff, except for the step of glycolysis, takes place. And so that's occurring deep inside of the mitochondria. And it's actually a really interesting organelle because it has its own DNA. You can actually track maternal lineage through mitochondrial DNA. Some of you might know this already, but for the rest of you, you're going to learn something new here. So you can track paternal, so, father of father of the father of the father of the father of a person up the line through looking at the Y sex chromosome, because they always get it from their biological father. Well, you get your mitochondrial DNA. And if you're thinking, why does the mitochondria have DNA? That's a good instinct to have. We'll talk about that in a second. But the DNA in the mitochondria always comes from the mother because, well, let's be honest, there's really not much in sperm except for it's basically a bit of DNA with a jetpack strapped to it that just gets launched. It's not, in terms of cellular function, there's not much there. So the egg has to fill in the rest of the organelles and stuff like that. So it has the other half of the genetic information and a lot of the cellular mechanisms, including mitochondria, to get things started. So it's not coming from any of the paternal DNA. It's coming from actually the structure of the egg. So that means that if you're always getting the mitochondria of your mother, that means that your mother got mitochondria from her mother, and so on and so forth. And so you can track Y chromosomes and find out things like, oh my God, so many people on the planet have Genghis Khan's Y chromosome. Guy got around, his kids got around, those kids got around, everybody's got Genghis Khan Y chromosome. Well, not everybody, but a lot more than you'd expect. And you can track maternal lineage through mitochondria. So you can do the same thing with the opposite gender. As long as you don't have to cross back over, but like, the father of your great-great-grandmother, that's not going to work. But you can look at her mother, or you can look at your great-great-grandfather's grandfather. You can track it that way. And through, mutations and things like that, that's generally where you start to see splits, and you can find some differentiation there as well. They're not common, but that happens. So, why does the mitochondria have DNA? I promised you an answer to that question. I'm sure for some of you it was a burning question. And the answer is they used to be their own organelles. Or they used to be their own organisms, excuse me. They weren't organelles. So, early on, the planet's environment, so oxygen is very reactive. So it wasn't in the atmosphere. So O2 gas didn't really exist because it's very reactive. That's why we don't have just oxygen floating around in our bodies. We always have it tied to something like hemoglobin. We always have to have a chaperone for it. You don't want to let it loose because if it hits the DNA, you're going to get a reaction. So it's very reactive, which meant it's just not going to be around on its own, which seems weird because we're kind of really used to having it around now, right? We take a deep breath to emphasize that point. Like that's some oxygen right there. I would not exist without it. So it seems weird to think of it not being around. Well, guess what plants do? They make oxygen, right? They're the first form of life, was life that photosynthesized. Wasn't necessarily plants, it was like single cell versions, like little organisms that did photosynthesis. Not quite up to the level of multi-cell plants necessarily. And they started to change the composition of the atmosphere. So there was 0 O2. And then there was 1% O2, and then 2% O2, and then 3% O2. And at a certain point, it meant that O2 was going to be in the cell, even though it was always trying to get rid of it, just because there was so much in the environment. And that actually started causing oxygen poisoning of these organisms. They didn't have a way of dealing with oxygen, and so it would damage them and they would die. And the very first organisms to come up with a way to metabolize oxygen and neutralize it so that it wasn't harming them was a group of organisms called peroxisomes. And so these peroxisomes could do reactions with oxygen and keep it from harming their cellular environment. And so you've got a couple options when you're those other cells. You can come up with the same option as peroxisomes. You can develop that as well. And it's all, random, by the way, right? So you just randomly stumble across it. And then, boom, you have it. Or you invite your peroxisomes over to hang out and deal with your oxygen. And so what happened was these peroxisomes started kind of coexisting within larger cells. And from that point, it was essentially the entire movie of WALL-E. The entire thing was just like people got on a ship that took care of them and they became fat and useless. And that's what mitochondria are. So those people in WALL-E, compared to people today, are like what mitochondria are compared to peroxisomes, millions of years ago, or billions of years ago, or I don't know, 10,000 years ago, according to whatever. Just kidding, those people don't acknowledge peroxisomes. Anyway, however long ago it was, they had these out in the wild, fending for themselves, go-getter peroxisomes, and then they got into larger cells and just sort of mellowed out and became mitochondria. So they're just an organelle now. But they still have that DNA as a reminder of when they were in their prime, when they were taking, they were working for their meal instead of just having it handed to them. And so that's why they have their own DNA, because they used to be their own organism. And now they're just an organelle like any other. I mean, they still have, not necessarily, like they do a lot of important stuff. And their structure 100% feeds into how they do their job. So now what they do is they're essentially handling oxygen, by the way. They're still handling oxygen. The oxygen end of respiration. But they're making ATP doing it. And so this inner membrane, all of this is occurring in the mitochondria, but within this inner membrane we have an area where we're constantly pumping out H+ ions. So we have a bunch of H+ on the outside and just a little bit on the inside here. And so to run these pumps, we use NADH, we use FADH2, and we just use these electrons, we just hand them off, and we get more and more work out of them until eventually we make water with them. We use H+ and oxygen, we make water. And so we pump these H+, and you think, well, why don't we use them to just make ATP? Well, the answer is, this is just how it developed. This is what we do now. So it's a really kind of, duct tape together system, because we pump these H+ and then they force their way back through. They always want to go from high to low concentration, so they come back through, and when they do, they power a protein that's essentially just a turbine and a press put together. And so it takes ADP and phosphate, and using the force of the H+ going through it, because this is the only way back into the inner membrane for the H+, that force is used to crunch together ATP. So we just run these pumps. That's the first half. And then the second half is we have ATP synthase, is the name of the protein, creative as always, that synthesizes ATP by letting H+ back in a controlled route. So it's kind of like you have, water behind a dam. You let some water through the dam, and you can generate electricity from the force of that water flowing. Well, through the force of the H+ flowing, we can generate ATP. And I have a video that I have linked below these. It's not my own. So I have linked a video below these. I highly recommend it. It's a 3D visualization of the whole thing. It's the Harvard X video for the electron transport chain. It is, some name like that I have there. I forget what I typed exactly. And you get a cool voiceover with a cool accent, which is more than you ever got from me. So you're welcome. You get to experience the Harvard lifestyle for about, I don't know what it is, 10 minutes. And it will show all of this in action. And so I'll have a thing at the end here where I talk about just the highlights, because I'm giving you way more information I expect you to know. But if we zoom in a little bit on this process. There's a protein that handles NADH, there's a protein that handles FADH2, and then they hand off through an intermediary. So these electrons get pushed in here, boom, we pump protons. They get handed to the Q here, that carries them to 3, and then 4. 2 handles FADH, and then it pumps to 3 and 4, and every step of the way we're moving H pluses, and we're never letting those electrons on their own. They're always being chaperoned around because they are, if O2 is reactive, these are super reactive. So you don't want loose electrons in a cell. So they're being handled carefully. And the overall chemistry is very simple. 2 H pluses, 2 electrons, oxygen, you get a water. But what we're really doing is we're really using that energy from this reaction to pump H pluses, just so we can let them come back through a controlled area. And again, check out that Harvard X video. It has a really good, check out specifically the ATP synthase part of it, if nothing else, because it's really cool to see this thing in action, because it really is. I mean, it looks like a mechanical device. It is a press that just rotates, and it just presses together phosphate and ADP, ka-chunk, 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 and it makes ATP. And it's powered by H pluses flowing back in. So we're always pumping them out, and then we're letting them back in and making them do some work along the way. And we pump them out, and they come back in, and we do some work. And so before I get to the highlights there, the last thing I'll talk about are these. There was a chemical that was in these old diet pills from like the 1800s, dinitrophosphate, or DMP. And what it would do, so it would work in terms of ramping up your metabolism, but with one key side effect that I'll talk about in a minute. What it would do is it would allow H pluses to cross this membrane without going through ATP synthase. So it kind of attack the integrity of this membrane. It would kind of, imagine it was like punching holes in the wall. And so there were holes in the dam, water was flowing through. It wasn't always powering our turbine. Sometimes it was just going downriver. And so you only have like 30 seconds of ATP. So you're constantly, this is constantly happening, whether you're awake or asleep, you're on a jog, or you're sitting and you're watching TV or whatever. This is going on. This is going on as you watch this video right now. If it's not, that's one less grade for me to put in at the end of the quarter, I guess. But anyway, we're making ATP. And to do that, we're pumping H pluses, and then they're coming back. Well, if you don't have enough H pluses on the outside compared to the inside, you got to run these pumps harder. You got to pump faster. So if the H pluses are able to just come back in, well, you got to pump them back out, and they come back in, you pump them back out. Now, if they come over here, you get ATP, but sometimes they just go through. And so you would take these diet pills, go to sleep, and you'd wake up in the morning, I assume just completely sweating, because you would just be running hot the whole night. Because it would be like you were, working out, because you were just burning through all your carbohydrates, all your fat, like you're powering these pumps, powering and powering and powering. So what is the side effect? Well, if you take a minute and you think about it, it might be clear what the problem is. We have to have H pluses go through here or we die. Because we have to make ATP or we die. So if you affect the integrity of this membrane, and you don't have enough H pluses going through here, the side effect is death. And there's a lot of factors that would go into that dosage. And it wasn't like there was an FDA back then. There was no approval process. There was none of that. And even today, the allowed side effects are going to be directly proportional to the disease itself. And so there are ways to lose weight without a potential side effect of death. And so that's going to be what the medical field is going to promote. If it was a cancer remedy, okay, if you might die from it, well, maybe if you're in stage 3 and it can possibly bring you back, or stage 4, it could possibly bring you back, that's an acceptable risk. But as the risks of the disease get lower, the allowed risks of the treatment get lower. And so be clear, I'm not like indicting this process at all. It makes complete sense to me. Like, I am not going to risk death because I'll just go for a walk every day or whatever, and be actively involved in becoming a puddle of sweat instead of just having it happen in my sleep, because then at least I know I'll wake up in the morning, and that's worth it to me. And if you're wondering, does this stuff get used for anything else? Well, it is used in rat poison today. It's not the only kind. There are other kinds of rat poison. Let me be clear. Don't start taking rat poison as a diet pill. Don't do that. I'm not encouraging you to do that at all. In fact, don't ever touch this stuff. But you can use it to kill rodents, and I don't encourage poisoning them in general, because they're going to find the worst place to die. It'll be like in the deepest recesses of your house, and you get to play What's That Smell for 3 months while you have a decaying 5-pound Norwegian giant rat somehow in your wall. Or it gets out, instead of going deeper, sometimes they leave and then they get picked off by a bird and that dies, or a cat or a dog or whatever. Just kind of like killing your way up the food chain. So, yeah, just do the traps. Alright, so, you never know what advice you'll get. So you got rodent control advice, you're welcome. It's actually pretty relevant in Eugene these days, I would say. Most people I know end up having to deal with field mice and stuff here. So here's the takeaway. Talking about the electron transport chain, I talked about all kinds of stuff. Rodent control will not be on the final. And the diet pill stuff won't be on the final. That was just for your information. Same with DNA of mitochondria. That was just kind of like for your information. So you should understand that, one, mitochondria, they're built to do this. This structure is designed to generate ATP. That is what they do. And they have these two membranes to do it. They're both necessary. That allows us to create an area where we have a bunch of H pluses, an area where we don't. So we get a gradient. And then we create that gradient by burning through NADH and FADH2. So all the stuff with respiration we're talking about before. It might have seemed like NADH and FADH2 are kind of throwaways. Like, oh, we're making CO2 and we're doing this and we're doing that. Oh, we've got acetyl-CoA. Well, it turns out that actually this is what we're doing. We're making this stuff and then we're using it to make ATP in the mitochondria. And so they power those pumps, and then we have that gradient, we have that area of high concentration and low, and so the H pluses try to go back from they're getting pushed into the area of high concentration, and they want to push their own way back into the low concentration. And so they do that through ATP synthase, which uses that force to crunch a phosphate and ADP into ATP. So that's kind of the takeaway here. And that's it for the course material. So don't forget the final is cumulative. And please take a moment, fill out the course evaluation at the end. I'm always looking to improve things. Every time I teach the class, I want to be better than the last. My apologies to those of you that have taken the class with me the first time. Always some bumps in the road there. And I hope you've enjoyed the quarter. And good luck with the final. If you have questions, as always, just ask. And hopefully I've given you a nice strong foundation where you can launch into anatomy and physiology next quarter. And and just a heads up, like that, those are tough classes, by the way. They are going to ramp up from this one. I had a student once that was mad at me because I didn't make that clear enough. So let me be clear. A&P is harder than this. Alright, now you know. I'm sure you knew already. But you can't be mad at me. I told you. It's always tricky. Like, I don't want to discourage you guys. But get ready. Alright, so, yeah. So good luck with that. Good luck with the end of the class. And, yeah. Enjoy that online learning. And, if there's anything you can think of that I can do to help out students in the future, I'm always open to advice. Let me know. And if there's anything I can do to help you out, with any of the older material, doesn't have to be the new stuff, let me know, and we'll figure something out. All right.